So her her title is cell type specific m6A programs underlying immune evasion. So Michelle achieved her bachelor's degree in Capital Medical University in Beijing in 2007. Then she started to pursue her PhD degree in the Institute of Biophysics, Chinese Academy of Science, where she studied how myeloid cells are involved in the immune surveillance of tumor. Since 2013, Michelle moved to Department of Pathology, University of Chicago, to start her postdoc research with Dr. Yang Fu, where she's still working on how different myeloid lineages work in the tumor immunosurveillance. During her both her PhD and her postdoc research, Michelle has demonstrated several major discoveries in how myeloid cells played a role in the tumor immunosurveillance. For example, in one of her work, she demonstrated the CD47 immunoblockade. The effect of CD47 blockade actually depends on T cell and is mediated by classic dendritic cells. Since 2017, Michelle started her own lab in School of Medicine, Tsinghua University. In her own lab, Michelle worked on how different RNA modifications plays a role in tumor immunosurveillance in both myeloid cell set and tumor set. So let's welcome.
Okay, thanks more for the great introduction. Also, thanks LA and Tran for organizing this great e-meeting. So I'm going to talk about the recent discovered m6A program. This is a new RNA modification very broadly on the mRNA, and how does this program work in different immune cells? So because all of you are very senior immunologists, so I will bring more background of the RNA modification. So there's more than 100 different types of RNA modification in mammalian mRNA. But for the most abundant one, this is a so-called m6A. And this is A could be methylated by the writer protein. This complex called -3, -14, and WTAPR. And this is what I'm going to talk about today, about how does the -3, -14 complex regulate the macrophage transition into microenvironment. And the other way, if you want to get rid of m6A to become a normal A, then you need an eraser, then we call it the methyltransferase. And there's two identified the methyltransferase FTO and OCT5. And another scenario I will bring to you this about the methyltransferase. How does it control the tumor immune surveillance or immune evasion in the in solid tumor? Because m6A is most prevalent, and it also this has unique distribution on the 3' UTR on the mRNA. Also, this process is dynamic. So recently, there's a lot of paper has been demonstrated its important role in physiological process or in the pathological process. For example, there's a paper in the stem cell culture system or the naive to prime culture system talk about the role of -3, -14 in directing the pluripotency of stem cell. Also, in 2017, Richard Sowell, Huabing, they published a Nature paper talking about how the -3 control the T cell homeostasis. So all of this evidence supported it might be important for mammalian cell to maintain the m6A level or use the m6A program to adapt the environment or the stress. Because how does the next question, how does the m6A modification can be functional translated? There's a lot of radar protein can bind and recognize m6A modified sites. And then the consequence of such recognition could be diverse. For example, if they recognize by, for example, YTHDF2 or DC1, it could degrade the mRNA in the nucleus or in the cytosol. Or another radar protein recognized m6A could mediate the splicing. And in 2015, people realized that if m6A could be recognized by YTHDF2, this could trigger a cap-dependent translational acceleration. That means m6A also could promote the translation efficacy of certain mRNA. And when we first initiate to our initially determined to step in this field, the first question for us is it also important in the tumor development or the tumor initiation? For us, we care more about the how does microenvironment has been orchestrated and how does this different type of cell adapt to the microenvironment and change their function will also be important for the m6A or the cell could utilize m6A to somehow respond to certain stimuli or change the half-life or translation efficacy of certain mRNA. So with this question, we somehow search a lot of literature, and so far this review talk about the role of m6A in the tumor setting. For example, in hypoxia state, somehow the aquifer or the methyltransferase could protect the tumor or prevent the tumor initiation. Also, the other case, there are other methyltransferase and metal-3 or metal-14 could promote the tumor metastasis or the tumor AML development. So if you look at the function of m6A, you will wonder why they play opposite role in the tumor cell, and is it also the case in the immune cell? So our basic idea is if one cell, the cell A, or we think they in the state A or in the status A, they utilize the transcriptome. Somehow they can modify this through their methyl m6A program, and then to transit instead into another transcriptome B. And then somehow this cell state could permit its long-term adaptation in the tumor microenvironment. If we look at the transcriptome or the gene regulation module, you will see there's a dramatic switch between two states, but the cell lineage will not change. So we think maybe m6A methylation program in tumor cell is could be initiated or utilized or hijacked to manipulate the immune context and then avoid the immune recognition. But we don't know which writer, or we don't know is it true or which writer or which complex is involved. So before we start this, we somehow do a correlation analysis. So here in this figure, we utilize a so-called CTL score. That's a recent Nature medicine paper published by Shirley's lab. They somehow select several genes only expressed by cytotoxic CD8 T cell, and then calculate the average expression level of this 5 genes. So this so-called CTL score. So we correlate the CTL score with the expression level of different epigenetic regulator, including the regulator involving demethylation or histone modification or the m6A modification. So there's 2 published DNA methylation and histone methylation and histone acetylation protein they already know to mediate the immune escape in tumor cell. But if we look at the m6A methylation, we know or we observed, we found that FTO, this is demethyltransferase that responsible for the transition from m6A to A. So this FTO expression in a high level, then you will have less CTL score. That means there's a negative correlation between FTO expression and the CTL score or the CD8 infiltration. And this is in the melanoma. Now we look at the TCGA data set and also examine the other type of solid cancer, including the breast cancer or glioma or lung cancer. You will see there's also a significant negative correlation with the of FTO and the CTL score. So we think there might be a link between the FTO expression and the CD8 infiltration or CD8 cytotoxic capacity. So to detect or to evaluate whether FTO could mediate T cell or prevent the T cell infiltration, so we first choose 2 different type of cell. One is a hard tumor with very high immunogenicity with over antigen, and also a cold tumor. You'll see there's a Louis lung cancer with very few mutation. So we knock down the FTO in this 2 type of tumor, and we check the m6A level. You will see there's more m6A in the mRNA because the FTO is methyltransferase. So of course, if you knock down FTO, there's more m6A. But if you culture this tumor cell in vitro, you will see there's no difference in the cell proliferation. That means FTO in tumor cell does not affect the basic proliferation, aggressive proliferation of tumor cell in vitro. Also, you put this tumor cell in the rat mice, that mice don't have a T cell or B cell. And if you look at the tumor curve, they also show similar pattern. That means this tumor intrinsic destruction of FTO does not affect the tumor growth. But if you put this tumor cell wild type or NC or the SH FTO into the syngenetic mouse, that's wild type mouse, then this mouse contain include T cell and B cell. Then if you look at the tumor curve, it hardly can grow out. And also this is the case in LLC tumor model. And we also check the tetramer positive or tumor-specific positive CD8 T cell within the tumor microenvironment. We look at if we knock down FTO, there's more CD8, tumor-specific CD8 coming in, and also the interferon gamma production as well as the JunB production also increased upon the FTO knockdown. So this data suggests that probably disrupting FTO in tumor cells somehow can inhibit the or promote the infiltration of the tumor-specific T cell. And this is also can be achieved in a very cold tumor model. So as we all know that the T cell activation is initiated by antigen-presenting cell, especially the dendritic cell. And recently, people use BAF3 or the XR1 DTR mice to get rid of or delete so-called migratory DC or the CDC1 then you will see less T cell priming. So we assume that probably if we knock down FTO tumor cell, it will somehow sensitize the dendritic cell to cross prime to CD8 T cell. So next we detect the cross priming capacity of the dendritic cell. To test this, we isolate the tumor draining from the dendritic cell, especially the migratory CD1 XCR1 positive DC, and then co-culture with the antigen specific T cell. And then we check by evaluating the interferon gamma projection, we can compare whether this DC shows enhanced capacity to prime CD8 T cell. And if we look at the dendritic cell isolated from tumor barrier mice in knockdown group, you will see there's no difference between knockdown versus control group in terms of dendritic cell mediated cross priming. So we somehow also check the percentage of tumor-infiltrating CDC and also the macrophage, and we saw there's no difference between knockdown or the control group. So we somehow rule out possibility that the dendritic cell is involved in such different T cell priming phenomena. And then before that, I'll also test the other possibility, because we know that the tumor cell, if T cell infiltrate into the tumor, somehow could immediately meet with or encounter with tumor or with APC. So we turn to dissect whether tumor cell directly suppress the cytotoxic function of the T cell. So we culture the knockdown tumor cell or the NC tumor cell with the CD8 tumor-specific CD8 T cell. And by RNA-seq, we can see if we knock down FTO, the T cell, CD8 T cell tend to express higher level of cytotoxic or effector gene like granzyme or interferon or perforin L2 and so on. And if we examine the interferon projection in in vitro culture system, we will see if we knock down FTO, there's like 5-fold increase in terms of the interferon gamma protection in CD8 T cell. And also the cytotoxic capacity of CD8 cell also enhanced when you knock down FTO in the tumor cell. So this means probably tumor intrinsic FTO could limit the activation or effector status of the CD8 tumor-specific CD8 T cell. Because the FTO is responsible for the demethylation of the m6A. So the direct mechanism could be FTO somehow change a group of mRNA and then sensitize the tumor cell to CD8 mediated killing. So we want to test which gene or which group of m6A modified gene is involved. Then we perform m6A-seq. If you look at the global transcriptional modification, there's a very unique or the conserved pattern of m6A on the 3 UTR. And then also, if we check the difference between this m6A level, you can see the knockdown up to they were increased m6A level, and also this is the exactly m6A gain region, showing there's more m6A gain in the FTO knockdown group. And if you look at this m6A gain region and look at this, which gene can somehow modify or changed, m6A level can be changed if we knock out FTO. Well, we found that there's a lot of transcription factors, especially some basic family, for example, gene or CBP, like contain the m6A site, and also when knocked down FTO, their m6A level increased. So we select several of these transcript factor and do the PCR to confirm this gene expression level, because there's one possibility that if you gain m6A, then you can be get degraded by the degradation machinery faster than the normal A. So we think probably if you knock down FTO, there's more m6A and there's more chance to expose to the degradation machinery, then you have less mRNA. And then we check the mRNA level of this m6A modifier or the m6A gain gene. You look at the gene CDBP or JunB, this three transcriptor belong to BZ family. Their expression level or mRNA level also reduced as we expected. But how to prove this is this such downregulation could be is attributed by the exact m6A site on this gene. So we development where we construct the DCAS13 fuse FTO. This DCAS13 system is like a DNA or Cas9 on the DNA setting, but this the Cas13 is to bind RNA. So we fuse the Cas13 with FTO and then direct by the specific guide RNAs, and you can get rid of the m6A on the specific site, loci, especially on the detected m6A gain site on JunB or CBP. And if we look at the Cas13 system fused with Y-type FTO, if we get rid of m6A, right, there's we assume there's this RNA will be more stable, and then the expression level should be enhanced. And as expected, then we get rid of the m6A on the m6A gain site. CBP expression level is increased. And if we use the DCAS13 fusion with FTO mutant, then there should be, then we didn't see such an increase in terms of expression. And we also check the protein level. If we get rid of m6A, the CBP and JunB and this two gene protein level can be also enhanced. So this above figures show that if we target specific m6A gain sites, you could somehow get rid of m6A. We can somehow prolong the enhanced mRNA level and also increase accumulation of this BZ family. So above we can confirm m6A modified site recognized by FTO, especially this BZ family could be modulated by FTO. Then we still cannot connect there such alternation of BZ family to the phenotype we observed previously. So we next also checked the C gene of JunB, CBP, this BZ family, whether they can target the SDR transcript factor. We assume that they probably bind to the different DNA loci and then promote the transcription initiation of different genes. So we wonder whether the downstream of this BZ family could be directly explained the phenotype of the tumor cell in the CD8 culture system. So then we perform a toxic. If we look at the knockdown versus control group, we will see there's a lot of chromatin peak has been lost their accessibility. That means their genome will tend to be closed. And this last region, if you look at their function, their or their nearby gene, you will see most of them are involved in the glycolysis. And then we pick up the several glycolysis-related enzymes. We will look at the several enhancer sites can be relatively close when you knock down FTO, and this site could be recognized by the JunB, C, and CBPb, and these are the published data, published ChIP-seq data that predict this is an enhancer of the glycolysis enzyme. So there's we somehow trying to connect the phenotype with the FTO modified basic family, and we found that probably this basic family can be enhanced in the FTO. Can be downregulated in the absence of FTO, and then such downregulation lead to the relatively close accessibility of the ATAC signaling, and then this less accessibility could be reflected by the downregulation of the glycolysis related enzyme. So to validate whether it is the case, we also check the expression of the glycolysis enzymes. We'll see that there's a lot of enzyme can be downregulated if we knock down FTO. And also if you use the Cas13 use of FTO and get rid of m6A on the CBP or JunB, then you promote the CBP and JunB expression. Then you can also upregulate the expression level of glycolysis associated enzymes. So next, we wonder whether such downregulation of the glycolysis pathway could be reflected by their glycolytic capacity. So we perform seahorse assay. This control group, we will add glucose and adenomycin to boost its glycolytic capacity. You will see enhanced signal of ECAR, but if you look at the SHFTO group, their enhance is relatively weak than the NC group. And if we do the mass spec to detect the metabolites in the cytosol or in the tumor culture system, you will see there's less glycolytic metabolism in the FTO knockdown group. So this all of this data confirm our hypothesis that probably when you disrupt or the knockdown FTO in tumor cells could limit the glycolysis capacity. And then probably this is the reason why clinical culture with CD8 T cell, this CD8 T cell response or the CD8 activation score can be enhanced because they all relies on glycolysis, and there's a competition between tumor and T cell. So to test whether we enhance the glycolysis, whether there's a less CD8 T cell response, and then we use the oligomycin. In this assay, if you add oligomycin, there's somehow will further improve the glycolysis. So we add oligomycin FTO group, and then we culture this tumor cell with the CD8, and then you will see without oligomycin, of course, knockdown FTO will sensitize the T cell capacity to recognize tumor. But if you enhance the glycolysis, the interferon and the granzyme production by CD8 cell are reduced. And then this also we also overexpress this JunB in the knockdown group. And then if you look at this this bar column, compare with SHO mark group, if it overexpress JunB, somehow they also downregulated the interferon production. But if you not overexpress JunB, also block the glycolysis, this such reduction can be rescued. So all of this somehow point out a very complicated or the long logic axis that disrupt FTO in tumors somehow could restrict the glycolysis and also sensitize the T cell recognition or T cell activation. And because there's no published, there's a very recently published cancer cell paper by Dr. Cai Guangyang's group. They use another molecule to treat AML in by disrupting the FTO activity in AML, and somehow they modified the previously selected inhibitor and then modify into a more soluble and with higher bioactivity novel molecule called DACT51. So we asked them if we can try their inhibitor in our system. And so this is their affinity. If we look at the DACT51, that they can reach to 400 nanomolar. And also if we treat tumor cell with the DACT51 and we compare the m6A gain region, they are perfectly recapitulate what we observed in the knockdown group. So that means this drug would be perfectly potently inhibit FTO and with very low off-target effect. So when we get this inhibitor, we first treat a different tumor cell with DACT51 and also confirm JunB and C and CDB expression. You will see we treat with 5 micromolar, and then this 3 gene BC family can be downregulated. And also if we treat SHNC group with DACT51, in comparison with the SHNC, we'll see after DACT51, also their ECAR capacity or the glycolysis capacity can be downregulated, and they could be recapitulated in our metabolites release. Also, we next we treat DACT51 in wild type tumor barren mice, and treat mice with DACT51 with only 3 dose, very low dose IP treatment. We will see the tumor tumor grow very slow, and also the CD8 T cell response can be enhanced exactly like we observed in the FTO knockdown group. Also, if we combine this DACT51 with PD1, PD-L1 antibody blockade, we can also see there's a synergenic effect. All of the tumor can be regressed. And when we revised the paper, one of review asked whether this drug can also show the potency in the patient-derived OPNAL or the PDC. So we collect the patient-derived OPNAL from the colon cancer, lung cancer, and then treat this PDO with DACT51 and check the basic family expression. And we found that this 3 transcriptor can be downregulated, like we observed, exactly like we observed in the mouse model, and also the interferon production by human-derived CD8 T cell also enhanced. So in this study, we somehow understand if tumor with higher level of FTO and then start to modify some, get rid of the M6 level on several basic family, and this family could upregulate expression level and trigger the glycolysis. And when the glycolysis capacity or efficacy can be accelerated in tumor cell, then they have very higher advantage over CD8 T cell and to consume all of the glucose and then somehow inhibit the CD8 from their recognition. So this way, solve this novel mechanism how M6 regulate the immune evasion. So with this question in mind, we also can assume another scenario. It is whether the immune cell in the tumor microenvironment, let's say our lab focus before is the macrophage, and we know macrophage somehow can switch between immune supportive to immune suppressive under different environment cues or stimuli. And such reversible programming could be modified by previously identified epigenetic modifier or the transcript factor, but we don't know how does the regulation module can be established and whether tumors like tumor cell immune cell can also utilize such program to establish its immune suppressive function and also on demand somehow protect the tissue from the insult of the T cell. So we propose another hypothesis. Actually, these two stories we start simultaneously in the same time. So we don't know which one work, but finally both of them works. So initially we ask very naive question is when naive immune cell, especially focus on tumor-associated macrophage, they can receive different stimuli when if they under different condition or facing the different environment cues, they can somehow quickly modify their M6A level or they methylate this M6 level and then immediately change the half-life or translation of their some functional related specific transcript, and such in timely and fast regulation program could be guarantee this type of cell could effectively reprogram this phenotype or its function. So we think it's a fast responding system and also it could be very cost effective because you don't need a lot of ATP to translate tons of cell, but you only need to get rid of one methylation and then change the whole consequence of the mRNA life. So we perform a very simple so-called experimental M1, M2 polarization system. I guess a lot of labs have done this before. So we somehow treat macrophage with interferon plus LPS or the L4, and we detect the M6 level in different stimuli. We will see there's a dynamic increase when you stimulate with interferon plus LPS sphere, but somehow the M6 level tend to downregulate in the L4 stimulation condition. So they somehow support our initial hypothesis that if they face different stimuli, they are able to change the M6 level. And then we want to move this whole system into a tumor-associated macrophage because M1, M2 just an experimental macrophage polarization system is not similar as what we isolate from the tumor tissue. So, but at that time, we don't know what is the subset of macrophage. We only know the some classical marker of macrophage. So we decide to carefully dissect the macrophage population within the tumor microenvironment before we detect their M6 level. So to achieve this, we inoculate LLC. This is a myeloid enriched tumor model. And then we isolate CD45, also including the T cell. But I told you before, this is a cold tumor. So most of CD45 positive are myeloid derived cell. And there are very few DC in this system. And if you look at the population of myeloid subsets, there we can briefly divide into three populations: monocyte, or the other two types of the macrophage, cluster 1 and cluster 2. And this is the signature gene expressed by three populations. And if you look at the macrophage, the MAC2, this green population, they express very high level of C1q, and also the MHC class 2, and the other are macrophage population class 1, they express high level of PTGS2. And so from now on, we call this C1q macrophage and PTGS2 macrophage. So the first, we want to test whether they differentiated into two different distinct directions. Because some people think macrophage can gradually, continuously differentiation along the same direction, but in a different state. But the other point or the opinion is they stem from the same. They start from the same point, but somehow they diverge into totally distinct lineage. So our data support that there's a very different, even unique direction flow between two subsets of macrophage. There, the differentiation trajectory could be very diverse. If you look at the diffusion map, you will clearly see there's two different differentiational trajectory along from the monocyte to the MAC1 or MAC2. And if you look at the expression, the different expression gene, you will see C1q macrophage versus PTGS macrophage. They have very unique transcriptions. And then we will carefully examine their DNA or histone or the RNA methylation, whether they have any preference rely on DNA methylation or on the DNA modification. And we also find that along the monocyte to C1q macrophage, if we compare with monocyte to PTGS2 macrophage, we will see the gradually gain the m6A methylation program, especially the methylated program. This is the -3 metal-14 can catalyze the m6A methylation. Also, the another radar protein, YTHDF2, that can mediate responsible for the RNA degradation of m6 mark gene. And we also confirm this phenomenon by staining, flow cytometry staining. And then because we have single cell, we know exactly which C1q, whether C1q express in the signature or the signature markers. So we select CX3CR1 MHC class 2. And in this double positive population, they express very high level of C1q, and the other population don't have C1q expression. And if you look at the -14, we think this C1q can be co-expressed with the -14 expression. But the another subset, macrophage subset, hardly or seldomly express -14. So they're confirmed that the C1q and the -14 can be co-expressed and along the monocyte to C1q macrophage differentiation. So we think this is a very unique distribution pattern or the expression pattern in the C1q macrophage. They have unique RNA methylation. Probably they have a unique RNA methylation program. And also if you look at, there's also another radar protein, YTHDF2, we construct the knock-in mice to detect whether this is the case in the double positive population. And also we found that similar to what we observed in the metal-14 expression, YTDF2 also highly expressed in the C1q positive macrophage. And all of this prove probably this population could utilize m6A to do something. But before we perform any functions that were knocked out, we confirmed the population we isolated are the exactly same population we detect by the single cell RNA-seq. So we somehow isolated double negative, double positive population, and then perform the bulk RNA-seq, and then reflect all of the differential gene detected by single cell RNA-seq. And we found that the population we isolated are highly enriched of some gene, especially or the previously expressed by the C1q macrophage we detected by single cell RNA-seq. So this figure demonstrates that if we use this two marker to we briefly separate C1q and PDGS macrophage, it does work. And then we from the next slides, several slides, when we perform any function assay, we usually isolated double negative versus compare double negative versus double positive. So next we isolated to detect their M6 level, because the double positive one, this is a C1q positive macrophage, have higher methyltransferase expression. So when we detect M6 level, they indeed have higher M6 level in comparison with the PTGS2 positive macrophage. And then if we want to dissect their function of C1q macrophage versus PTGS2 macrophage, we found the C1q positive macrophage, they have a higher expression level of macrophage activation or antigen presentation or response to interferon gamma, while the other PTGS2 macrophage, they tend to regulating angiogenesis or response to hypoxia. So this means they are somehow play a distinct functional role, as we saw in the differentiation trajectory. And then we look at the cell-cell interaction between the macrophage with CD8 T cell, because we assume that the antigen presentation or response to interferon are the hint that imply probably this macrophage tend to crosstalk more with the CD8 T cell, but the PTGS2 probably keep encounter with the other like structure cell or the vessel cell, and to keep the tissue homeostasis. So we compare their potential in terms of crosstalk with CD8 T cell. So all of this ligand receptor pairing or list are the potential ligand expressed by macrophage or the CD8 T cell, and then the circle, the size of circle means the expression level, and this color bar means whether there's significant interact with each other. Okay, and then if we compare the PTGS with C1q, we will see there's a lot of ligand receptor interaction between C1q and CD8 T cell, but this type of interaction will not be detected in between PTGS2 macrophage and CD8. So there's somehow imply or suggests that C1q has the potential to crosstalk with the T cell. And before we move to the knockout system, we also want to confirm whether the fluctuation of m6A level in macrophage is also detectable, or could be recapitulated in the colon or the human cancer patient. So we collect the several colon tumor sample from colon cancer patients, and we try to get rid of the epithelial or the tumor cell, and we call all of the right cell are the stromal cell. But most case stroma cell means there are macrophage and some fibroblasts. And if you look at the m6 level of every patient, if we compare their adjacent normal stroma cell versus the tumor stroma cell, the m6A level are downregulated. And if you look at the metal-14 expression on stroma cell, you will see there's a positive correlation between metal-14 and m6A level. That means in some patients, they somehow downregulated metal-3 level, probably because they don't have enough metal-14 expression. And also then we confirm that this metal-14 expression are sometimes detected or in most of the macrophage, but they express in a different level. In certain patients, the expression level is very low. So we want to mimic the clinical setting in the mouse model. We want to somehow delete or downregulate metal-14 in macrophage, and then we will assume this downregulation could change the tumor microenvironment. So we cross the metal-14 flux mice will LYZ cray mice. Also cross metal-14 with the MAFB cray mice to double check this is a macrophage specific deletion. If you look at the metal-14 expression, they are highly expressed in the macrophage, but if we knock out metal-14, there can be largely reduced, but metal-14 does not express or in a very low level in monocyte or the neutrophil. Then we detect or monitor the tumor curve of the knockout mice versus wild type mice with different type of tumor models, and get a similar conclusion that metal-14 knockout in macrophage indeed promotes tumor growth. And if we also knock out the other radar protein that are highly expressed by C1q macrophage, we will see the similar trend of accelerated tumor progression. And then we examine the myeloid cell, the distribution or the percentage of myeloid cell. We see there's no difference between macrophage monocyte and as well as the C1q or the PDGS macrophage level is comparable, and DC is also comparable. So they rule out there somehow you change the you knock out metal-14, you have somehow affect the other myeloid population. And then we look at the CD8 T cell in the knockout mice in F10 or the LLC tumor model. There's also a sharp reduction in the percentage of CD8 infiltration. Also, if you delete CD8 T cell, somehow the proof this such tumor controls is the CD8 dependent. So the next question is want to dissect which CD8 T cell in our knockout system is could be modified by the macrophage. So this is a single cell distribution map of different CD8 class subpopulations. You will see there's two distinct groups, and if we somehow calculate the co-expression gene model, we will see these are three major co-expression gene model. One is we call they're highly expressed exhaustion related models. So here we mark by the light blue, we call the exhaustion module, and also the co-expression gene enriched for the proliferation related gene, we call the proliferating module, and also the progenitor or memory or stem cell-like module, we call the progenitor module. And if you look at this 3 module, you will find there's CD8 T cell tend to enhance their proliferation score, and also the exhaustion score. That means with the infiltrating into tumor cells, somehow they accelerated their proliferation when they recounter with the antigen, and also the corresponded exhaustion score was also elevated. And we put this score and into a different project this score to the UMAP. You will clearly see which population have more progenitor score and which population is in a more terminal differentiated or so-called exhausted like or dysfunctional like state. We saw there gradually loss of a progenitor score, but also again the exhaustion or the proliferation score along the differentiation of CD8 T cell. And if we compare the wild type with knockout, if the major difference is here, there's a progenitor like population is missing, but there's a more terminal differentiated or the exhausted like state T cell accumulating the knockout mice. So we check the T cell function. There's less interferon or granzyme production, but also there's a so-called inhibitory receptor expressed higher in the knockout mice. Also the PD-1 tox double positive T cell could be the terminal differentiated or dysfunctional T cell. Their number also increased in the knockout T cell. So far we confirmed when knockout melanocyte macrophage change or shift of the tumor infiltrating T cell towards a relative dysfunction or terminal differentiation state. And next we examine the link between macrophage, multi-14 macrophage and such CD8 T cell phenotype. So because we mentioned before the C1q, they have more antigen presentation and response to interferon gamma signature. So the first idea is whether they have if you knock out melanocyte, they have less antigen presenting gene expressed, or the interferon stimulating response can be dampened. So we compared this gene in 2 different tumor models, and there's no difference. Also, rule out the possibility of ISC and also antigen presentation. And we also check the other node pathway, including co-stimulate signal 2 and also PD-L1, arginase CD206, MHC class 1, class 2, they all show similar express pattern. So that time we somehow stuck there. So we examined our m6A sick and also the differential gene expressed by the M14 knockout macrophage. And we found that if we knock out melanocyte, the M6 level can be reduced. Also, there's a minor relative downregulation of the M6A non-M6A modified gene in terms of their transcription level. And then we zoom in to look at the gene with M6A marker also can be upregulated in the knockout group. So there's a lot of ligand expressed by tumor-associated macrophage. And so we assume probably this ligand could be responsible for the CD8 dysfunction or the CD8 exhaustion. So this is several we mark here can be upregulated with the knockout. So their M6 level can be reduced after the knockout. So this EBI3 gene if we look at their expression level in knockout mice, they can be enhanced also in the DF2 condition knockout mice. And the previously study in 2007, the recent cell immunity and the immunology paper, they publish and then they know the EBS3 compare with P35 to form IL-35. And usually people dissect this protein, the Treg. If you knock out the EBS3 in Treg, somehow the Treg cannot suppress effector T cell proliferation. So that means that somehow the EBS3 positive, the expression of EBS3 in Treg could suppress the effector function of CD8 T cell, Th1 T cell. And then we wonder whether it's the case in our tumor model. So we block, neutralize the EBS3. We look at this. This is a white type mice, and this one is knockout, mitotic knockout mice. If you use the mitotic plus neutralizing antibody, somehow they can slow down the tumor growth. And if you use this mitotic plus EBS3 neutralizing antibody, and then sometimes delete CD8 T cell, then the tumor can also grow out. So they demonstrate that if you are not neutralize the EBS3 knockout, it can rescue the CD8. But if we further delete, and such effect is CD8 dependent. And we also confirm this by the interferon gamma projection, the knockout plus EBS3 blockade. So until now, we prove that block EBS3 can the selective blocking EBS3 selective regulation on the T cell function can protect or they rescue the most impressive impact of the mitotic knockout macrophage. And also we confirm this phenomenon in different setting of patient samples. There's a public data we download from melanoma or the lung cancer. And also you will see there's a C1q macrophage here. Here's a C1q march here. And if you, here is C1q macrophage. If you look at the monocyte differentiated into the C1q macrophage, you will see gradually gains DF2 or metafotin. Also this case in melanoma, but you will see the very opposite correlation or opposing expression pattern of EBS3 and mitofotin, right? If you have a higher EBS3, there's not much mitofotin expression, but along the differentiation when mitofotin accumulated, there's no EBS3. So we somehow think that could be very important in the tumor-associated macrophage. Also, we further confirm that macrophage with EBS3 expression, also with lower mitofotin expression, can be surrounded by in a close location with CD8 T cell, or the in contact with the CD8 cell. And this CD8 cell, but in a single cell RNA-seq, we will check, or we confirm that this cell are similar or express similar signature gene like TOX or PD1 or TIM3. And also this population increase along with the EBS3 expression in macrophages. So there could be also proof that our observation in tumor in mouse tumor model has some clinical relevance with the patient sample. So this is a summary of the whole study. We think the macrophage could utilize m6A methylation to enhance the EBS3 and then orchestrate a very immunosuppressive microenvironment and shift the T cell away from effector stage and to establish a T cell dysfunction, favoring microenvironment. And there's a lot of pity or in our design or study because we the initial, we want to dissect the immunosupportive module versus immune suppressive module, but at the end, we only find EBS3, and there's only very few a gene with m6A level can be regulated or downregulated or upregulated upon M14 or metal-3 knockout. So there's another question is whether there's some other regulation pathway can coordinate with the transcription. How does it achieved? We still don't know, and whether there's a transitory phenotype, because we only detect one time point at the one tumor model, so we don't know whether this is the dynamic shift from X or to Y module, and whether there's another nascent RNA involved. How does they degraded this whole picture we don't have, and we think it is very necessary to be dissected. So this is a study, the two studies accomplished by our graduate students, two graduate students in Tsinghua, and also the TAM stories done by Li Hui, and also postdoc in our lab. And the FTO stories collaborated with Dr. Yang in Shanghai, and also the Dr. Han in BIG. And that's my talk. I'd like to take your question. Thanks.
So any questions?
Hey, Shimon. This is Xiaolei. So really beautiful talk and so many interesting data. So I think I have two questions. The first is maybe a more general question. Is there any spatial and temporal regulation of these writers and the erasers of the m6A modification to prevent like futile cycles of like writing and erase?
This is a very good question. I think the answer is yes. And this also related to another question, the specificity of or preference of this writer or reader protein, how this can be achieved or determined, right? So I guess there's a two direction can be evaluated. The first one is people realize that the thermal transcript factor when they start the transcription initiation, there's the transcription rate is different. Somehow the code slow or faster transcription, right? So the slow pattern, somehow they can have more time to recruit the writer protein and then to deposit m6A on this mRNA. And that's also relying on the other coordinates of his modification or the transcript factor to achieve. And the other way we can dissect is the structure of the mRNA, because in the cytosol somehow they are mature mRNA, right? They have different the different lengths of 3 UTR, different structure of UTR. Somehow they can be selectively recognized by different radio protein or the different protein complex. I think but we have done that yet in our system. Yeah, but this.
Okay, yes, that's great. So that kind of alluded to my second question, which is on the FTO. So basically, FTO can like modify like almost like all mRNA, like including mRNA, tRNA, and like snRNA. So is there any substrate specificity has been found for the FTO?
I think they are everywhere. You're right. Somehow they can recognize every substrate, but in a different way, I guess, because somehow we found that if we modify the FTO or get rid of FTO, not every cell, or no, sorry, not every transcript change their M6 level. And somehow they can be compensated by another dimethyl transferase ACB5. So right, yeah, I think I hope I have the answer, but I don't know.
Okay, that's interesting. Yeah, I think that we probably
and also somehow there has some buffering system in this. I don't know what is. Sorry.
Okay. So maybe a related like a question is so I find out, so I just look up the database. It looks like the FTO is really highly expressed in ciliated cells. So is there any specific function like for FTO in these ciliated cells? Oh, it's not known.
Right. I think it's unknown still.
Okay. Yeah, because it looks like it's really like stand like out of the rest cell types. Like if I look at the cell atlas, I just wonder if there's any like specific physiological function for these FTO proteins. Because I think they play a role in like regulating fat, and obesity as well. But that's, I think, a really fascinating proteins. Yeah, thank you so much. Thanks.
So yeah, Chen wondered, did you try FTO clip-seq in your system?
Oh no. Yeah, because we did several times. We use several knock-in or FTO antibody, but the same our PAR clip does not work well. I don't know why. They usually work well in our other system, but FTO, no. We did, but we don't get the satisfied result. I mean, the quality is very poor. Yeah.
Michelle, I have questions about the signaling for the FTO. So you proved that FTO actually influenced the glycolysis in the tumor, and then it's influenced the CD8 T cell function. So my question, I guess, is that the exclusive signaling pathway? In other words, if you block the glycolysis in the tumor and then apply the FTO inhibition or the knockdown, can you still see the same effect, or you have additional effect?
You mean block glycolysis in FTO knockout tumor?
No, right? I think the first thing is, my question is this a linear signaling pathway? In other words, the FTO is only affect the CD8 T cells through the tumor glycolysis, or there's other effect besides glycolysis in the tumor?
Oh, okay. I have to say we have done try our best to enumerate all of the expected mechanism, including interferon pathway or the other pathway might affect the CD8. But in our hand, we feel desperate to finally focus on glycolysis because we didn't detect any difference on other pathway. But we cannot rule out there. Yeah, you're right. There probably another possibility can also affect the T cell function.
I guess my point is, what if you let's say block glycolysis in the tumor? I'm sure that time T cell will get affected. But if you on that base, if you suppress, you use your drug inhibition or you knock down FTO based on the glycolysis inhibition, whether you will see further activation or further influence on the CD8 T cells. compared with only glycolysis blockade?
I guess you mean this figure. This one, right? Oh, yeah, we don't have the NC with oligomycin. This is the sorry. So this one, sorry. This the only one we block the glycolysis well to DG. This is analog, and but unfortunately we don't do the in the FTO knockdown or with the drug treatment.
Okay.
Right.
Okay. Okay. Thank you.
So I saw Yuefeng raised hand, and then next will be Zhou Xu. So Yuefeng.
Okay, great talk. Thank you. And I have a question still related about the specificity of these erasers. So if you knock out another eraser, for example, LKBH5, can you still see the similar defect on the glycolysis on the tumor cells?
Yeah, we also knock out RB5, but the sound seems like the glycolysis does not change much, but they somehow also sensitize the tumor cell to CD8 medium killing, but probably through the elevated expression of mHC peptide complex. But we're still working on this right now.
Okay, so I think the question is these are not only have cell preference, right? For they work in certain type of cells, but also they have a preference for certain transcripts, right?
Right.
All right, thank you.
So Zhou Xu.
Hey, Shimon, this is a really interesting talk. Thanks. I have two questions. One is a general question. The other is more specific. For the general question about m6A modification, do you know in the FTO knockouts, in terms of the percentage of modified adenosine versus general pool of adenosine, how much increase do you observe after FTO knockdown? And then is that, do you know if the modification tend to be concentrated on like individual molecules or just more generally spread out across all mRNA species?
Sorry, I somehow skip your first question.
Yeah, just in the FTO knockout, what's the percentage of modified adenosine in the general pool? How much more increase do you tend to observe?
Let me see. The only way to quantify is QQ. We didn't show here. I guess it's only 20%, 10 to 20%, not that dramatic as we detect it for FB5 knockdown.
So based on some of the data you showed, it seems like the percentage of modified adenosine is about less than 1%, about 0.5% of the overall adenosine that are available. And in terms of mRNA molecule populations, how many percentage of mRNA molecules or transcripts do you imagine are modified with m6A?
I forget that number, but there's a lot of mRNA in terms of the if you think their M6 ratio can be downregulated like 1-fold or 2-fold, there could be half of the mRNA.
I see. And in the first part of your talk, it's really interesting. You showed this really nicely that in the tumors, even if you knock down FTO without CD8 T cell, without T cells, they still grow in the same way within the mice. And also in vitro, there's a drastic difference in the metabolisms of those tumor cells if they don't have FTO. There seems to be a decoupling between their cellular metabolisms and their cellular proliferation. Do you think the m6A modification on mRNA is kind of like a balance to make sure their metabolism is coupled with cellular proliferation?
I guess in vivo somehow, right? It could be. But we don't know whether they rely on other metabolite pathway besides a glycolysis. So somehow could be compensated by other. We have checked the glutamine or the other metabolism is somehow relatively upregulating. We knock down FTO. But whether they coupling with the proliferation now, I don't have answer. Yeah.
Thanks.
So if we don't have more questions, let's thank Michelle again for the wonderful talk. So
Thank you.